Well, thank you for the introduction and good afternoon, everyone. I'm excited to share my image today and the work behind it entitled Journey Through the Mind's Maze, Nanoparticles for Targeting Brain Cancer. Glioblastoma is a highly aggressive brain tumor that is notoriously difficult to treat. The first treatment strategy is usually to surgically remove the tumor. However, because glioblastomas are often highly diffuse and integrated into the healthy brain tissue, even if most of the tumor can be safely removed, there are often many tumor cells left behind that can then lead to recurrence. One treatment strategy that's being developed for treating glioblastomas, particularly those that are difficult to remove with surgery, is a direct local infusion of therapeutics into the brain. In this case, rather than going through the bloodstream, therapeutics must penetrate a dense network of pores between the cells in the brain, which in this image is represented as the red pathways. Each of these gaps is about 100 nanometers wide, which is about 100 times smaller than the smallest blood vessel in your brain. In the Hammond lab, we design nanoparticle drug carriers that are designed to penetrate this system and specifically target drugs to cancer cells. We've engineered these nanoparticles to be about 100 nanometers in diameter so that they can freely flow through these gaps, but slowly enough that therapeutics are retained for long enough in the brain to have their effect. The objective of my research in the Hammond lab is to learn how different nanoparticle surface chemistries interact in the environment of the brain. In these three brain tissue sections, the yellow to red gradient is showing the distribution pattern of three different nanoparticles of similar diameter, but with different outer surface coatings. By designing the right surface, we can dramatically increase the distribution, enabling coverage of as many tumor cells as possible. Using a different technique known as flow cytometry, we can then analyze the cells in the brain and specifically isolate the cells that have taken up nanoparticles. And that is shown by the red dots in the inner box there. If we investigate this cell population further, we found that several nanoparticles enable increased uptake by tumor cells, which is shown in the red bar, and decreased uptake by healthy cells in the gray bar, which aims to improve the safety of this technique. If we zoom in at a much higher magnification, we can see this process of nanoparticles penetrating this dense cellular structures in the brain. The yellow to red gradient is showing the trajectory of nanoparticles as they travel away from the infusion site, between the cells with their nuclei shown in dark blue, and the cell bodies of the most abundant cell type in the brain, astrocytes, shown in cyan. This research was made possible through the support of my research advisor, Professor Paula Hammond, and the entire Hammond lab, specifically those who worked with me on glioblastoma projects. I'd also like to acknowledge the excellent core facilities here in the Koch Institute and thank our funding source.